Hello and welcome back to our discussion of gas transport. In this video, we will be talking about oxygen transport. Okay, so far we've talked about how we get gases from our blood into the atmosphere, into the air, and vice versa. But now we're going to start to talk about how those gases, once they get into our blood, how they are carried in the blood, starting with oxygen, okay? So oxygen is transported in the blood in two ways. It's carried as dissolved gas in the plasma, and that's less than 2% of our oxygen in our blood is carried that way. And then the other way that oxygen is carried in our blood is bound to hemoglobin. So that is most of the oxygen in our blood. So over 98% of the oxygen in our blood is carried bound to hemoglobin. Okay, so I want to start by reminding you that oxygen has a low solubility in water. And since plasma is mostly water, that means that we can't carry very much oxygen as dissolved gas in the plasma, okay? So our oxygen demand by our tissues greatly exceeds the oxygen carrying capacity of the plasma. Therefore, we need some other way to carry oxygen in our blood. Okay, the solution to that problem is hemoglobin. So I want to start by doing a little bit of review, see if you can answer these questions. Where is hemoglobin found? How many oxygen molecules can bind each hemoglobin molecule? So go ahead and think about that for a second. Okay, so hopefully you remember hemoglobin is found in the red blood cells, okay? And each hemoglobin molecule can bind 4 oxygen molecules. So I just want to remind you, hemoglobin is made up of these 4 protein subunits. Typically 2 of them are alpha chains, 2 are beta chains. Each of those protein subunits holds a heme group, and each heme group can bind 1 oxygen molecule. Therefore, each hemoglobin molecule binds 4 oxygen molecules. We can describe the binding of oxygen to hemoglobin using this chemical equation right here, okay? This symbol we refer to as oxyhemoglobin. This is hemoglobin bound to oxygen, okay? Taking a look at this equation, hopefully you remember your equilibrium principles from chemistry. I would like you to consider this question. Given a fixed amount of hemoglobin in the blood, what determines how much oxyhemoglobin is present? So take a moment to think about that. Okay, so if we have a certain amount of hemoglobin, then the thing that's going to determine how much oxyhemoglobin we get is how much oxygen we have, right? So if we have a lot of oxygen, that is going to drive the equation in the rightward direction, and it's going to favor the formation of oxyhemoglobin. If we have a low O2 concentration, what's going to happen is we're going to favor the dissociation of oxygen from hemoglobin, okay? This is physiologically relevant because, as we covered in the last portion, we have different partial pressures of oxygen at different locations in our bodies, okay? So just to remind you, in the alveoli and in the arteries, we have a high PO2 of about 95, 100 millimeters of mercury. In the tissues and the veins, we have a lower PO2 of about 40 millimeters of mercury, okay? These differences are going to drive the binding and the unbinding of oxygen to hemoglobin. So let's take a look at that in cartoon form here, right? So remember, this is our equation, okay? If we look at the alveoli, we have a high PO2, If we compare that to this equation, are we going to favor the forward or the backward reaction? Take a moment to think about that. Okay, so this should favor the forward reaction. We're going to favor the formation of oxyhemoglobin, which makes sense functionally because what we want to do is we want those red blood cells to pick up that oxygen so that it can be transported in the blood. If we look at PO2 in the tissues, that's 40 millimeters of mercury, kind of low. And we're using up oxygen in the tissues, right? So we're depleting oxygen, which means that this equation, what is going to happen to it? Is it going to go forward or backwards? Think about that for a second. Okay, so hopefully you figured out it's going to go in the backwards direction, which makes sense because we want to at the tissues, we want to dissociate oxygen from hemoglobin so that we can free up that oxygen for the tissues to use, okay?